Tzdaka, probably pronounced it wrong, but I apologize for that. The Volcano Institute in Israel, the effect of juvenile hormone on the activity of viruses in bumblebees. Please, the floor is yours. The remote is there. The green arrow, please, in front.
So my name is Assaf Tzdaka. I come from the Hebrew University and I'm a master's student at the Lab for Behavior and Diseases. And today I'm going to present you my project, the effect of juvenile hormones on the application of the Israeli paralysis virus in Bombus terrestris. So let's begin. It doesn't work.
Here you go.
Now is the timer, please.
It's working.
Backward is forward. Oh, so I'm getting...
Okay, this is very confusing.
I'll get this too. So, we all know that bees are essential pollinators for agricultural yields and food security. The most common two species are buff-tailed bumblebee, which is Bombus terrestris, and the one that we all know, the European honeybee, which is Apis mellifera. And like every other creature, those bees are prone for several diseases, and the deadliest amongst those diseases come from viruses, and there is a deadly virus which is called the Israeli acute paralysis virus, in short IPV. It's an RNA virus with a genome size of 9.5 kilobases. It causes a systemic infection in bee colonies, and it has a high mortality rate among infected bees, deadly, as I said. So what do we know? Let's do some virus and hormone interaction. So what do we know? We know that viruses are obligate parasites, right? They must have a host in order to reproduce. Also, viruses' application depends on the host's metabolic rate. And we know that hormones alter cellular activity. Look, it's been proven. Juvenile hormone functions as a metabolic rate accelerator in bumblebees. It's from 2021. So what does juvenile hormone does in bumblebees? So it regulates reproduction, metabolism, and hibernation, we will later see. An increase of the GH levels results in development of the ovaries in worker bumblebees and also the queens. And also, the same increase causes an acceleration of the metabolic and protein synthesis rate. Look how cute. I snapped a pic of a queen bumblebee incubating her youngs, and these are the eggs. And remember this motif because it's gonna stay with us. JH is low in queens before hibernation, but it's high in queens after hibernation. So, in order to better understand that, let's look at how juvenile hormone interacts with the bumblebee queen life cycle. So, we are in the late fall, it's the love season, virgin queens are mating, and believe me, male bumblebees are great lovers. And the queen bumblebee, when she's young, her JH levels are low. And then she goes to the second stage. In this stage, in winter, bumblebees hibernate underground, still with what did I do? Oh. Still with low JH levels. And then we move on to the third phase. The queens emerge, they begin their solitary phase, JH and metabolism levels are starting to rise in the spring, and when we follow to the summer, they reach to the highest peak when the colony is developing, which is the social phase. Which leads us to the research question. Does juvenile hormone affect the production of the Israel acute paralysis virus in Homo erectus? Let's see. So we have hypothesis. We know that before hibernation, JH is low, and thus also metabolism is low. So we are expecting to see low replication of the virus. But when JH is high, after hibernation, we expect a high replication of the virus as well. So what did we do? We took queens, we raised them in small plastic cages, we fed them, we grew them, we held them in a controlled chamber, and then we gave them a treatment by infecting them with either living or dead virus. 7 days after that, we collected the bees, we performed tissue extraction, and we collected 3 tissues, fat body, ovaries, and brain, and afterwards, we proceeded through a qPCR analysis in order to check the levels of the virus in those tissues. So, for first experiment, we took queens before and after hibernation, in which they have like natural hormone levels. Before hibernation, low JH, and one group received live virus, the other one received the dead virus. And then we took queens that are after hibernation, high JH levels, and again, same thing, we gave them live virus and dead virus. And what did we see? So first of all, you can see here the graph. Each bar represents a different treatment group. And because we are dealing with viruses, we transformed it into a logarithmic scale. The differences between here, and here are huge, okay? So here we see queens before hibernation with dead virus, and of course, low virus levels. Here we see the queens before hibernation also with dead virus, not a lot, but let's focus on those 2 groups and compare. We have here bees before hibernation and after hibernation. Both received live virus, and we can see in the fed body that those with the live virus after hibernation got significantly more virus. And the picture is the same, almost the same in the ovaries, and the same direction follows in the brain, but in a less significant way. So we can conclude that the IPV replicates significantly more in the fat body and ovaries of queens after hibernation in comparison to queens before hibernation, but less in the brain. We know that in terms of physiological differences, there are many parameters that could create this picture if we look at queens before hibernation and queens after hibernation. So we wanted to isolate. What if we isolate just the JH and see if this one is the parameter, the jackpot? Let's see if it works. So what did we do? We took queens which are all before hibernation, which means they all have naturally low levels of the hormone. And again, now we gave them a treatment of DMF solvent, which is a solvent of the JH hormone. One group got live virus, and the other one got the dead virus. We did the same, but this time we added JH treatment. And again, one group received the live virus and one group received dead virus. And what did we get? Look how beautiful. Same picture in the fat body, in the ovaries, and the brain. The treatment group, which was treated with the JH plus live virus, look how significant the difference is. So we can say from that, we can conclude that from that, the juvenile hormone increases IPV replication in queens before hibernation if we give them the treatment. But that's not all. We thought to ourselves, what if we take queens which are after hibernation and they have naturally high hormone level, and we add, in addition to that, in an artificial way, again, juvenile hormone. So we repeated the same method of the experiment, but this time with the queens after hibernation. And what did we get? Surprise. No difference between the group of DMF and live virus, and JH and live virus. So in this case, juvenile hormone treatment does not affect the application of IPV in twins after hibernation in all 3 examined tissues. So let's have a small summary. We saw that IPV replicates at a high rate in twins after hibernation compared to twins before hibernation, in the fat body, in the ovaries, but less in the brain. And the JH treatment increases the replication of the IPV in twins before hibernation in all 3 examined tissues. But when we take twins which are all after hibernation with naturally high JH levels, we get an effect of sealing. It doesn't affect them. So the big conclusion is juvenile hormone increases the application of IPV in bumblebees. But why would IPV interact with JH activity? Let's suggest a mechanism in terms of evolutionary view. So remember this life cycle of the bumblebee? So let's look at it again. Here is the queen hibernating, and then comes the IPV virus and attacks, and then it replicates. Oops, dead end. The bee dies, the virus dies. But what if the virus replicates when the JH and the metabolism are at their highest peak? Oh, what a good way to replicate. Viruses love this. They love when the cell is busy with lots of activity, so the virus can replicate inside the mother, the queen bee, transform into their daughters, and from her daughters who are foraging, they will serve as a taxi for the virus to spread itself further on to a local and further populations of other species. That's it. I want to thank my supervisor, Dr. which is over there, to my other supervisor, Professor Mochkanovsky, my colleagues at the lab, Elad Bonda, Izik Sadeh, Omer Kama. Special thanks to Dr. Victoria Sroger and her student, PhD student, Tallin. Thank you.
Yeah.
Questions?
Other questions, please. Please here. And then there. Yeah. Maybe you second, you first, right? Because I saw your hand first, I'm sorry, yeah?
Maybe I missed something, but what would be the mechanism of the juvenile hormone action, the molecular mechanism on increasing the possibility of virus infection? What's
The connection is if juvenile hormone serves as a metabolic accelerator, it makes the cells work harder, use a huge amount of machinery, and the protein turnover will increase, and it will provide the viruses with all the parts it needs in order to reproduce and replicate itself, hijacking the host cell. We don't know exactly what is the molecular mechanism of it, but this could be the next.
Okay, so it's not the problem of the virus invading the bee. It's not the problem, it's not the effect that the virus has the chance to invade the bee from outside, but it's somehow replicate itself, the virus that is already inside the bee, right?
Yes, it could be like in a latent stage, and when the JH activates, yes.
I want to ask you if you also analyze the levels of juvenile hormone in bumblebees.
If I analyze the exact amounts?
Yeah.
No.
Yes, another question here.
In your experiments, you show what happened when you inject the virus into the body of the bee. The injection itself has its own effect. What would you say that will happen if the bee will feed on this virus, like in the same system, without the injection? And because also the virus should go through some challenges throughout the tissues of the bee when it's not injected but ingested by feeding?
Okay, that's a good question. We took that in account, and when you inject a bee with a virus, you need to use a very small amount in order for it to activate itself because it's bypassing all the defenses, right? Like Varroa does with honeybees. And based on other articles, we know that when you feed bumblebees, you need to use a much higher quantity of the virus in order for it to come into action. But we wanted to make the timeline shorter, so we used the injection. But we guess that in nature, when they get the virus when they get infected, it takes a much the time span of the disease is much longer, and maybe it could be latent. That not in our experiment, the virus eventually killed the bees, but when they get the GH levels. But in nature, we suppose it stays latent and works in a much moderate way. It keeps its host live, and also it manages to reproduce without killing it entirely. So there's a difference between lab and nature.
Thank you. So I would have also a quick question. Kind of juvenile hormone has so many functions in a honeybee or in insects in general. It triggers so many different molecular mechanisms and functions. So in your study, do you believe it's really juvenile hormone itself what is having effect on virus, or is it some proxy of some kind of physiological process what it's mediating or what it's regulating?
That's a good question. We also took that into account. We need to remember that JH is an upstream effector, if you might, which it activates so many parameters, and we need further to investigate which one exactly, but we know that it's the switch that activates the process.
Okay.
The main switch.
Thank you. I would like to thank all the presenters in this symposium again today. And we will continue tomorrow 9 o'clock. I believe it's 9 o'clock, right? 9:30, sorry. Tomorrow morning we will continue our symposium here. And now go and enjoy coffee and the posters.
אני אקבל אותו די עם הסרטון, הכל ברצף. כבר 18 דקות ברצף.
אתה צריך לחבק אותו, להגיד לו מה זה. שהוא מגיע, הוא מצלם אותך.
הוא עצר שם אצל הוויקטוריה.
נו, מה אומרים לו הישראלים נחמדים האלה?
אתה מתכוון אצלך. אצלי הוא מקבל חיבוקים. כפרה עליו. קבלו אותו, קבלו אותו!
דן.
לא היית נראה לך.
שיחקת אותה, מה אתה אומר? איך היה?
זה רק היה משחק, אף אחד לא יודע.
איך היה?
שחקן טוב, היה נהדר. אני הרגישו בשקופית האחרונה
נהנית? רק שתדע שכל החלק הראשון גם מצולם, אתה יודע, שלא עובד לי, לא עובד לי.
באמת? נהדר, אני שמח, זה מצחיק. זה רק יקרה לי, כמובן.
כן, עכשיו אנחנו כבר ב-19 דקות.
וואי.
איך עושים? אי אפשר לעבור לסלפי.
אי אפשר, אפשר לסובב את